Good morning, student. So today we will be studying another topic, that is tissue culture. Okay, so as traditional breeding is a technique which was not capable of producing, or which has been failed to keep pace with demand and to provide sufficiently fast and efficient system for a crop improvement. So for that, the people are not getting the demand as per the increasing population. So certain approach has to be taken by the certain experts. That means a crop को इम्प्रुभमेन्ट के गर्नु पर्यो, बढाउनु पर्यो। किन भन्दाखेरि चाहिँ ट्रेडिसनल ब्रिडिङको कारणले गर्दाखेरि जुन चाहिँ डिमान्ड र छ त्यसको पिसलाई चाहिँ फुलफिल गर्नु सकेन। त्यसको कारणले गर्दाखेरि चाहिँ another approach, another technology start भयो, another technique start भयो, that is known as tissue culture. Okay, so it is a technique. Tissue culture is actually a technique हो यो चाहिँ to grow, or it is a technique of growing plant cells. यसमा plant को cell लाई grow गरिन्छ cell बाट, okay, tissues बाट, organs, and कस्तो conditions मा भन्दाखेरि चाहिँ in a medium of the sterile cultures under controlled aspartic conditions, and that medium will be controlled by the expert. So यस्तो conditions मा चाहिँ के गरिन्छ, start गरिन्छ. This tissue culture was proposed by Gottlieb Haberland, okay, in 1902. In 1902, he has shown his experimentations in the German Academy. Okay, in the German Academy of Science in the 1902, where he shows that his experimentations of the single cell. Okay, so the proponent of the tissue culture is Gottlieb Haberland. Okay, so this tissue culture means this will have a certain ability to generate a whole plant from a single cell, and that is known as explant, and that ability is known as totipotency. That means एउटा single cell बाट, एउटा single tissues र single organ बाट एउटा whole plant लाई के गरिन्छ, regenerate गरिन्छ, एउटा abilities हरु हुन्छ plant मा, and that a single cell is the explant. Okay, and it is called totipotency, and that potential is known as totipotency. Okay, so let's see here. Tissue culture refers to the isolations of cells, tissues, or organs from plant and growing them as particularly in suitable containers on an artificial nutrient medium under controlled conditions. That means the cell will be isolated from the whole plant, tissues or organs are isolate गरिन्छ र चाहिँ as particularly suitable containers मा राखिन्छ, अनि artificial nutrients हरु चाहिँ त्यसमा लगाइन्छ र के हुन्छ, certain control under the control of the expert in the laboratory, in vitro culture of the plant will occur. Okay, we have been talking about this explant. Explant भनेको चाहिँ, the explant is a small fragment of tissues, okay, which will be removed from an intact plant under the sterile conditions for the propagations of plant. That means explant is nothing but this is the fragments of the tissues, or this is the fragments of the cells from the plant, okay, so that this can be developed into a whole plant. Okay, so and this totipotency means, totipotency is the ability of a single cell to divide and to produce all of the differentiated cells in an organism. That means एउटा single cell छ, okay, which has been isolated from the parent, isolate गरेको छ। यो single cell मा चाहिँ एउटा यस्तो ability हुन्छ जसको कारणले गर्दाखेरि चाहिँ उ divide भएर okay, this will get divide भएर multiple cells मा divide हुन्छ, and ultimately एउटा whole organism मानिलिउँ एउटा plant मा developed हुने उसकोमा ability हुन्छ, and that ability is known as totipotency. Okay, so an explant is any part of the plant that is grown in a test tube under a sterile nutrient medium. So यो चाहिँ कुनै पनि प्लान्टको पार्ट हो जो चाहिँ केमा नि डेभलप गर्छ भन्दाखेरि चाहिँ कुनै पनि एउटा टेस्ट ट्युबमा चाहिँ के गर्छ, ग्रो गर्छ. Okay, the nutrient medium must provide a carbon. अब कुन मिडियम लगाउनु पर्यो, के हुँदैछ भन्दाखेरि यहाँनिर एउटा प्लान्टबाट सेल टिस्युज अर्गेन्स कुनै पनि कुरालाई एक्सट्र्याक्ट आइसोलेट गर्दैछ ओके आइसोलेट गरिसकेको त्यो पार्टलाई भन्छ एक्सप्लान्ट त्यो एक्सप्लान्टलाई चाहिँ अब टेस्ट ट्युबमा लगाउँछ अनि ग्रो गर्छ के सर्टेन न्युट्रिसन्सहरू चाहिँ त्यहाँनिर हामीले एप्लाई गर्नुपर्छ अब त्यो न्युट्रिसन मिडियम चाहिँ के रहेछ न्युट्रिएन्ट मिडियम चाहिँ भने कार्बन सोर्स सच एज सुक्रोज इनअर्गेनिक सेल्स vitamins amino acid and growth regulators like auxins and cytokinins. These are the hormones or these are the growth regulators which will help to grow the shoots on the roots all the stems okay in the plant यो सबै कुरालाई चाहिँ एड गरिसकेपछि now the explant will be developed into a whole plant body okay the method of producing thousands of plants in a very short time through this tissue culture is known as micropropagation अब हेर है एउटा सिंगल प्लान्टलाई बाहिर इन द इन्भाइरोमेन्ट फिल्डमा डेभलप्ड हुनुको लागि चाहिँ एकदमै टाइम लाग्छ तर इन ल्याबोरेटरी के हुँदैछ एउटा प्लान्टको बडीबाट टिस्युजलाई निकालिँदैछ सेललाई निकालिँदैछ अर्गेन्सलाई एक्सट्र्याक्ट गरिँदैछ आइसोलेट गरिँदैछ एन्ड इन द कन्ट्रोल्ड इन्भाइरोमेन्ट विदिन द ल्याबोरेटरी अर इन vitro means lab okay in the vitro this culture will be there these tissue cultures will be conducted in the laboratory in short period of time एउटा जुन चाहिँ प्लान्टको टिस्यु सेल्सहरुबाट चाहिँ thousands of the whole plant developed हुँदैछ and that culture is known as micropropagation okay So clear? This micropropagation भनेको चाहिँ के रहेछ micropropagation is the propagations of plant involving cells and tissue culture. So cell र tissues बाट developed हुनु plant tissue is the culture in laboratory to produce plantlets. Plantलाई चाहिँ laboratoryमा के गर्छ plantlets मा produced गर्छ. It is been used in orchids, okay, sugarcane, potato, gladiolas हरुलाई चाहिँ यस्तो तरिकाले used गर्छ. If you see the uses of this micropropagation then the number one will be it is a very productive technique for superior varieties यदि हामीलाई superior राम्रो खालको varieties चाहिएको छ भने चाहिँ we can conduct micropropagation from these micropropagations in a short period of time we can get a large number of plants okay it can be applied to interspecific hybrids if we want a hybrid of a different types दुईटा species को बीचमा दुईटा plant को different plants को बीचमा चाहिँ हामीले एउटा नयाँ plant बनाउनु पर्यो hybrid चाहियो भने चाहिँ that is also done through micropropagation okay inter means between two different species okay it is useful in cases where seeds are dormant यदि seeds हरु चाहिँ longer period of time को लागि चाहिँ dormant means inactive dormant means inactive okay dormant means inactive longer period of time को लागि inactive भएर बसेको छ भने चाहिँ त्यतिखेर हामीले we can extract the cells tissues and we can propagate the plant from this culture okay but there is a limitations everything will have its the pros and the cons so let's say the limitations यसको disadvantage चाहिँ के रहेछ भने it is not easily applicable in remote agricultural areas like ours area हाम्रो एरियामा चाहिँ यो चाहिँ के छैन एकदमै हामीले यसलाई इजिली पाउनु सकिँदैन okay so you can see in the picture this is the pictures of the plant tissue culture first of all in number 1 you can see that tissue sample is scraped from the plant को parent plant बाट tissues लाई cells लाई चाहिँ के गर्यो scrap गरेर निकाल्यो and then in a petri dish मा चाहिँ उसलाई राख्यो okay tissue sample placed in the agar growth so यहाँ भित्र एउटा medium छ त्यो medium के छ agar medium okay which will be consisting of a different nutrient which we have already discussed along with the cytokinins and auxins or growth regulators okay र यसको medium ma चाहिँ this plant like a tissues will be grown okay and this small samples will be developed into a tiny plantlets अब tissues बाट चाहिँ small plantlets हरु developed हुँदैछ अब यो plantlets will continue developing Okay, this will get the nutritions, and these plantlets will be ultimately planted into a compost. You can see that the leaf has been arise, the stems has been arise from these plantlets. Okay, so these plants will be genetically identical. Now these plants, okay, this the plant which is coming from the यो प्लान्टबाट एउटा ठुलो प्लान्ट अल्टिमेटली डेभलप हुन्छ हैन। यो प्लान्ट छ, सबै यो प्लान्टहरू चाहिँ के हुन्छ, जेनेटिकली आइडेन्टिकल। द्याट मिन्स एउटा प्लान्ट अर्को प्लान्टसँग जिन जेनेटिकली जिन लेभलमा नै के छ, आइडेन्टिकल हुन्छ, सेम हुन्छ टु द ओरिजिनल प्लान्ट। द्याट मिन्स यो प्लान्ट जुन चाहिँ नयाँ प्लान्ट बनिएको छ यो भयो, दिस वान इज द ओरिजिनल प्लान्टसँग जेनेटिकली दिस एन्ड दिस प्लान्ट विल बी लेटर अन आइडेन्टिकल। Okay, and they are called somaclones, rather identical, and the genetically identical plants to the original plant is known as somaclones. Okay, so this like a micropropagation is conducted in especially tomato, banana, apple. Okay, and through this process the whole plant will be developed. So tissue culture is also used for recovering healthy plant from the diseased plant. For example, एउटा कुनै पनि प्लान्टमा डिजिज लागेको छ अरे होल प्लान्टमा, र त्यो प्लान्टको एउटा यस्तो सेक्सन जहाँनिर चाहिँ डिजिज लागेको छैन, त्यो सेक्सनबाट चाहिँ टिस्युज र सेललाई एक्सट्र्याक्ट गर्यो अनि ल्याबोरेटरीमा त्यसलाई डेभलप्ड गराइ इन्टु प्लान्टलेट्स एन्ड आफ्टर द्याट प्लान्टलेट्स विल बी डेभलप्ड इन्टु न्यू हेल्दी प्लान्ट। ओके, त्यो जुन चाहिँ नयाँ बनिएको प्लान्ट हुन्छ द्याट विल बी डिजिज फ्री। Okay, and this meristem, which is free of virus from infected plant, is removed and grown in vitro to obtain a virus-free plant. For example, if the plant is suffering from a viral disease ले चाहिँ plant suffer भइरहेको छ भने plant को एउटा यस्तो पोर्सन हुन्छ जुन चाहिँ पोर्सनमा चाहिँ के छ, भाइरसले इन्फेक्ट गरेको हुँदैन, that portion is known as meristem. Okay, meristem चाहिँ इन्फेक्टेड प्लान्टबाट एक्सट्र्याक्ट गरेर बाहिर निकाल्छ एन्ड देन द्याट विल बी ग्रोन इन ए विट्रो, द्याट मिन्स ल्याबोरेटरीमा चाहिँ ग्रोन गर्छ। ओके, ल्याबोरेटरीमा इन ए पेट्रोडिस अर इन ए टेस्ट ट्युबमा ग्रो गर्छ र त्योबाट बनिएको जुन चाहिँ प्लान्टलेट विल बी अफ भाइरस फ्री प्लान्ट। Okay, so scientists have a cultured meristems of a banana. अरू कुन को चाहिँ मेरिस्टेम कल्चर गरेछ भने banana, sugarcane and potato. Okay, you can see that this is the tissue, okay, culture occurring in the banana. The scientific name of the banana is Musa species. This banana will have a different species. Okay, this is the Musa species. So you can see that this is the whole plant of the banana. बुझ्दैछौ, बनानाको एउटा होल प्लान्ट छ, यसलाई हामीले सोर्स प्लान्ट पनि भन्न सकिन्छ। फ्रम दिस सोर्स प्लान्ट द बल्ब कम अफ द सोर्स प्लान्टको जुन चाहिँ एउटा बल्ब कम छ त्यसलाई हामीले एक्सट्र्याक्ट गरिन्छ, यसलाई प्राइमरी ट्रिटमेन्ट गरेर चाहिँ यसलाई हामीले कन्टेनरमा के गर्छ, विल बी स्टोर इन अ कन्टेनर जहाँनिर चाहिँ रिप्रोडक्सन अथवा मल्टिप्लिकेसन विल अकर। Okay, after that multiplication, now from this bulb shoot and root will start developing. Shoot र root developed हुँदै जान्छ and then के हुन्छ, now primary hardening, that means shoot र root developed भइसकेपछि चाहिँ that okay, bulb will be separated into a different containers and that is known as secondary hardening. And after secondary hardening, that plant will be like planted into a different plants. यो कारणले गर्दाखेरि पनि के भयो, सिंगल एउटा बनानाको सोर्सबाट चाहिँ large amount of multiple numbers of banana plant we have extract. Okay, so next if we see, in case of the sugarcane, the scientific name of the sugarcane is Saccharum officinarum. Okay, and यो Saccharum officinarum that is sugarcane लाई चाहिँ कसरी टिस्यु कल्चर गरिन्छ भन्दाखेरि sugarcane को apical bud used as a explant. That means जुन चाहिँ apex means जसको shoots हरु leaves हरु आउने which will be projecting towards the sunlight पटि जुन चाहिँ project हुने एउटा part छ that is the apical bud भनिन्छ यसलाई चाहिँ extract गरिन्छ and this bud will be used as a explant. Explant means a segment or fragments of the plant. Okay, and this will be stored in a container में store गर्छ जहाँनिर यसको shoot tips आउनु थाल्छ. Okay, will be provided with a certain nutrient यहाँ provide गर्छ and then shoot initiate हुन्छ. shoot के भयो यहाँ अझै डेभलप्ड हुँदै गयो. ओके, shoot डेभलप्ड भइसकेपछि स्टार्ट भयो, यहाँ इनिशिएट भयो, यहाँ इलोंगेसन द्याट मिन्स अझै बढ्दै गयो. ओके, एन्ड इनिसिएसन्स अफ मल्टिपल सुटिङ, यहाँ सिंगल सुटिङ स्टार्ट हुँदैछ, सुट्स हुँदैछ भने यहाँनिर नम्बर्स अफ सुट्स हेभ बिन स्टार्टेड डेभलपिङ, एउटाबाट थुप्रो सुट्सहरू निस्किँदैछ. सुट मल्टिप्लिकेसन अझै प्रोसेसमै छ. After that, now the roots also start appearing. Roots पनि start हुँदैछ. त्यसको बादमा now this plant will be separated singly. एउटा प्लान्टलाई सेपरेट गर्छ र डिफ्रेन्ट कन्टेनरमा चाहिँ ग्रो गर्नु लगाउँछ. आफ्टर हार्डनिङ त्यसको बादमा चाहिँ this will be grown in a certain space. Okay, so single shootबाट चाहिँ large number of plant has been produced. So this is happens in case of the tissue culture of the sugarcane, that is Saccharum officinarum. Okay, so this one is for the potato. Okay, and the scientific name of the potato is a Solanum tuberosum. Okay, so first of all, in the figure A, you can see that this potato is like having a certain nodal cutting explant. Okay, harboring axillary bud. This will having a axillary bud start हुँदैछ. And in the B मा shoot initiation on start media. So के हुनु थाल्यो, shoots हरु चाहिँ start हुनु थाल्यो, certain shoots हरु developed हुनु थाल्यो. Okay, and now in number C, shoots elongation बढेर गएकै छ. Okay, and number 4, nodal cutting explant harboring auxiliary buds appears and shoot grown on media. That is certain this shoot will be cut and the further development of the shoots will occur, and this will be consisting of a certain medium जहाँ न्युट्रिएन्ट्सहरु लगाइराखेको छ. फर्दर सुट्सहरु के भयो, डेभलप्ड भएर जायो, मल्टिपल नम्बर अफ सुट्सहरु यहाँ डेभलप्ड हुँदैछ. ओके, एन्ड आफ्टर द्याट के भएर गयो, नाउ दिस shoot harboring auxiliary buds and microtubers. You can see that ट्युब्सहरु पनि स्मल माइक्रोट्युबर्सहरु स्टार्ट हुँदैछ. Okay, and this microtubers initiation from noble explant. यो microtubers चाहिँ कहाँबाट आयो, noble explant बाट आउनु थाल्यो. So after that चाहिँ this plantlets will be having a certain shoots such as 6 देखि 6 देखि चाहिँ 7 वटासम्म चाहिँ shoots र 5 to 7 centimeters को लेन्थमा आइसकेपछि चाहिँ यसलाई चाहिँ हामीले फाइनली पटमा ट्रान्सप्लान्ट गरिन्छ. Okay, so from a single potato, a large number of these potato plant will be like formed. So this will all happens in case of the tissue culture. Okay, so next there is one term called callus. Okay, so this is I think not there in your book, but this term is very, very important. And this callus means this is a callus tissue, which means an unrecognized proliferate mass of cell produced from isolated plant cells, tissues or organs where when grown as practically on artificial nutrient medium in glass vials under controlled experimental conditions. That means यो callus मा चाहिँ के हुन्छ भन्दाखेरि चाहिँ for example this is a callus मानिलिउँ जहाँ के हुँदैछ आर्टिफिशियल इन्भाइरोमेन्टमा चाहिँ यहाँनिर के डेभलप्ड हुँदैछ भन्दाखेरि चाहिँ अनरिकग्नाइज अर्गनाइजेसन मास अफ सेल प्रड्युस्ड हुँदैछ जो चाहिँ हामीले प्लान्टबाट एक्सट्र्याक्ट गरेको थियो त्यो प्लान्ट चाहिँ के भयो डेभलप्ड हुँदैछ. मानिलिउँ प्लान्टबाट हामीले यहाँ प्लान्टबाट सेल एक्सट्र्याक्ट गर्यो र हामीले यहाँ डेभलप्ड गराउँदैछ यहाँ मास नम्बर अफ सेल्स डेभलप्ड हुँदैछ विच इज अन लाइक रिकग्नाइज्ड एन्ड द्याट इज नोन एज क्यालस okay which will be like grown in a controlled experimental conditions जहाँ चाहिँ हामीले artificial nutrient हामीले medium of glass मा हामीले provide गरिन्छ that is known as callus tissues. Okay, so